Hello, please subscribe for more videos. This is Clinical Genetic channel. Today we are going to talk about mutation. Mutation can be occurred due to inversion, translocation and different mechanisms like non-selection. Today we are going to talk about first inversion. To identify and understand it better, I am using different colors here. In a chromosome, you know there are multiple genes and that is in a particular place of locus. You know what is locus? It is. Here I am putting different colors and to understand inversion, we are making some breakpoint here. In between the pink and the blue, there's one breakpoint, and pink and the green, there's another breakpoint. Now I am drawing it again by keeping a distance from the breakpoint, so you can identify it better. So when the chromosome get divided like this, then during inversion, the middle part turn 180 degrees. So the dark blue part will coming in down direction and pink part will be going in upper direction. I'll draw it again to you to identify it better. Okay, it will turn 180 degree. That is the inversion. So I'll draw. See, it changed, and now it fused together. So we'll find a new chromosome with the inversion. So you know, with this type of changes, the protein that going to synthesis will be different. Let's see it in future, in next step of video in the future. Okay, and the second part, that is translocation. For that I am using another picture here. This is a first we are doing two pairs of homologous chromosomes here. This is the first one, this second one. So in translocation, genetic material will get transferred from one chromosome to other. It first we'll see when one side of the chromosome is occurring here, chromosome change. Okay? If this is occurring in one side only, this is heterozygous translocation, because in the alleles only one part get translocation, so it's heterozygous. The both side is occurring, then homozygous translocation has occurred. So that's another mechanisms of having a mutation. First one we identify and understand, that is inversion. This is translocation. Okay, so by both this mechanism, patient can get a mutation. Okay, I think now it's clear to you. Okay, now we are going to this translocation as example like Philadelphia chromosome that also occur with the translocation. Okay, so those translocation inversion leading to multiple diseases. Other part of mutation change is by deletion. Deletion, even single nucleotide or maybe multiple nucleotide, that can also lead to multiple diseases. Okay, deletion for us to identify, sometimes it's not visible in karyotyping. Sometimes it needs further high technology to identify that. So why deletion is important? We know that in a mRNA, triplet of nucleotide act as codon. So the protein synthesis will be depend on that. If one nucleotide is deleted here, then what happens? Then the codon the mRNA will be reading will be, it will take the first two and skipping the deleted one, then it will take the next nucleotide. So what happened? You know there are 64 codons. Among them 61 is coding for amino acid and 3 for stop codon. If this new codon we synthesize, we identify now, if it is coding for a stop codon, then the protein translation will be inhibiting. So the truncated protein we will be having. If that part of the new codon is signaling for that among the 61 amino acid, then we will be producing a amino acid or protein. Okay. So if this is a receptor that is functioning well, for that receptor to function well, that need that configuration. So with the proper protein translation, we will have that receptor. But when the truncated protein is produced, they will have dysfunctional receptor. Sometimes even with the mutation, we are having normal receptor. How that is happening? Can you think of that? Yeah. That's the possibility is when there are 64 codons, among them we have 61 codons coding for amino acid. With the process of degenerate of codon, that means among the 64 we have 61 coding for amino acid, so only 20 amino acids are there. So what happened? More than one codon signal for particular amino acid. So we can still with the mutation sometimes have the normal protein. Okay, that's the end of this video. Thank you very much for watching this, and please subscribe for more videos, and thank you very much again.